a small object. Okay, so this is the question that was asked by this gentleman. Okay, so this is Arthur Ashkin, all right, and he basically asked this question and he started off experiments on this, all right, and these are some of the pictures which I have taken from his first paper, which I have put the reference over here, and it's a very interesting observation that he made. So you essentially have laser light which is being focused and sent like this. This is a cell which has certain very tiny particles, microparticles, all right. And what he found was that these microparticles were drawn towards the center of the beam. Once they were in the center of the beam, they actually started moving in the direction of propagation and then got held against the far wall of the cell. Now, how do we know that it was the light that was holding them? When he blocked the light, they fell under gravity. Okay, so it was obvious that it was light that was doing it. So then he could go ahead and make this kind of an arrangement, which are two counter-propagating laser beams, and then he could have a microparticle which was trapped in between. However, this was again not a very good trap because actually the particle could leave, from the sides. It was not a very great trap. So he decided that he's going to go ahead and design a very good optical trap for this. So what is exactly happening over here? So there are essentially two forces that are at work. The first force is the scattering force. So now, how do I explain it? So let me explain it in terms of, let's imagine this is an atom. There is incoming light. Now the atom, let us say, absorbs that incoming photon. It of course goes off to an excited state. Now, along with the energy, of course, there is momentum that has been transferred. So the momentum is going to be transferred in this direction. So this atom gets a kick in this direction. Okay, so it is all in the direction of propagation of light. Now this atom is not going to stay in the excited state forever. It is going to de-excite. Once it de-excites, it's going to send out a photon. That photon, re-emitted photon, could go off in any direction. It has no reason to go only in this direction or here. It can go out in any other direction. And so thereby, on time, if you average, you will find that the kicks which are coming from the emitted photons are going to be random, and thereby they are going to average out over time. But this, in the direction of the propagation, is going to be constant. So thereby, the atom is going to start moving in the direction of light. And this is, in fact, if you can transfer this to a microparticle, which is not the correct thing to do, but I'll explain how it works for a microparticle too. You can see that there is a reason for a small particle to move in the direction of light. Okay. Now, how about the other thing? All right. The other force, so let me just go to the next slide. Yeah. So this particular force, okay, can be basically put in like this. And here you can see there is something called as the time average Poynting vector, which is kind of depending on the amount of energy that is incident on the atom. And this is the scattering cross section. But now this is the scattering force. But let us understand what would happen to what we call as an electric dipole. So people who are not from physics, just a reminder that you must have studied this. But it's basically a dipole is nothing but the positive charge and negative charge being separated by a small distance. And even a neutral particle in an electric field, this can happen. So it can develop a dipole. Now dipoles in uniform electric fields usually will just align. However, if they are in an electric field gradient, they are going to feel a force. So now the question is, do we have an electric field gradient also, which is operational over here? And what we will find, okay, so this is just a lot of math, but let us just look at this. This is basically the dipole moment, as we call, and this will be proportional to the electric field. I have put a lot of things under the carpet. This polarization is not really a scalar quantity as it looks like over here, but let's not get into all of that. But this is a dipole moment, and the stronger the field, the stronger the separation, let us say, between the positive and the negative charges. And now the force is something which is proportional to the negative gradient of the potential. The potential is going to be the interaction between this dipole moment and the field, which is what we have given over here. And then if you take a gradient of this, you will find that there is an E dot E term comes over here, that is the electric field square, which is nothing but the intensity. So if you create a gradient in the intensity of light, you basically subject an electric dipole to a force. Okay, so that is what it means. So this of course is an explanation where the particle size is much less than the wavelength, and this is what is called as the Rayleigh regime. But you can now understand that a gradient in the light intensity can also subject an electric dipole to a force. So we have now come up with two forces. So let's now discuss how we are going to set this up. So a light source of course is required. So one can use a laser, all right. A huge advantage, monochromatic, highly directional, intense, okay. So we set up a laser, and the next thing is that you have to, so scattering force of course is going to be a part of it. Moment it is inside the light, there's going to be a scattering force. How do you set up gradients? So to do that, you firstly, let's think about how it looks in a laser beam. So a typical laser beam, okay, in one of the most common modes of operation would be like this, okay. So this is, you're looking along the beam direction. It is most intense in the center, and then the intensity basically falls off to the side. The intensity profile could look something like this. This is a function of position and central, and then maximum intensity at the center, and it falls off as a Gaussian, okay. So now what you realize is that obviously there's an intensity gradient in the radial direction. So that explains what Ashkin observed, that little particles were being drawn towards the central part of the beam. But now of course in the central part of the beam, what's going to happen is that the scattering force is also going to be enormous, because many more photons are going to be incident over here on the particle than over here. So as a result, the particle is going to then start traveling along the direction of the beam. Now you need to thereby create an extra gradient force which will counter this. So how do you do that? And that's very simple. Use something which will focus the beam, right? So you could use either a lens which focuses the beam or a mirror. This is the conventional choice, all right? And then you end up, as you kind of, collect the light and you focus it, you end up creating a gradient, an intensity gradient, and thereby you could also create a force in the axial direction, in the direction of propagation of light. So let us move further. And to do this, what do? You use an objective or a lens system which has a high numerical aperture. What's a numerical aperture? It's nothing but the ratio between the focal length, okay, the distance over which the light focuses, and the aperture which is collecting the light, okay, the diameter of the aperture which is collecting the light. So obviously, if this is an something like this, 1.2 or 1.4, what it essentially tells you that this focal length is going to be extremely short. It is going to be in fact shorter than the collection diameter, okay. So thereby you will have an extremely short length over which it is focused, and thereby you create extremely strong gradients. So now the question is, what does the force on the particle look like? So let's go on over here. All right. So this is, sorry, it's a bit grainy, this figure. So this is the axial axis, which is the line along which the beam is propagating. You can see the scattering force over here. The scattering force is of course going to be the most strong at the focal point, simply because intensity is largest, and thereby scattering events are the largest over here. And then this over here, the one in red, you see is the gradient force. Of course, it changes direction, because over here it will be towards the focus. Once you are beyond the focus, it's again going to be towards the focus. So the direction has changed. So the total force is going to be essentially this green line, and you can see this is the point where you find there is no force acting on the particle. So that is the position where it is going to get stably trapped. Okay, so this is how you would end up trapping a small particle using light. Now the question is, okay, this is what I explained was with atoms and dipoles and so on and so forth. Well, how does it work when we talk about, in the ray optics regime, when your particle size is much larger than the wavelength? What you can explain in terms of the refraction? Yeah, yes. Okay. Yeah, sure. Yeah, sure. So do you want me to, is there some doubt which you, all right. So okay, fine. I will try my best to go as slow, and if I'm fast again, kindly ask me to slow down. All right. So the previous slide, of course, sure. Okay, yeah. So, so let me just explain. So what you have over here on the y-axis is essentially the magnitude of the force which is being felt by the particle. Okay, so I hope everybody is with me. And here, this is basically the distance along the direction in which the beam is propagating. Okay, so this is the point where the focus is. Okay, this is the focal point. And here, of course, since you have focused it, the intensity of the light is going to be maximum over here. Everybody with me still? Yeah? So now what is going to happen, since the intensity is maximum over here, the scattering force, okay, is also going to be the strongest at this particular position. And that is represented by the blue line. As you move away from the focal point, what is going to happen? The scattering force is also going to come down in magnitude. All right? That is the first force. The second force over here, the one which is in red, is essentially your gradient force. Now the gradient force is always directed towards the focal point. All right? So it is basically what I wanted you to note is that if it is positive over here, if this part of the axis is positive, y-axis is positive, this part is negative. So what you have to understand is that this gradient force is always directed towards the focal point. Total force is going to be an addition of this red line over here and the blue line. So this sum is being shown in this green line. Okay. And what happens is that, see over here, at this point you can see the red is negative, the blue is positive, and they together add up to make a 0 point. Now a 0 point would mean this is a position where there is no force acting on the particle. So the particle can remain stably trapped over there. That is all I meant. Okay. So can I move on to the next slide? Right. So this is now how I would explain the situation for, let us say, a much larger particle. Okay. So this is, let's say, a large particle. Now please note the depth of the color. Okay. How dark it is shows you how intense it is. So obviously it's very low intensities over here. Here the intensity is extremely high. Okay. So now let us imagine there is a particle which is sitting a little off the center. Now what is going to happen? There's going to be, so the first thing I should mention to you, of course, over here is that the refractive index of this is higher than that of the surroundings. Okay, so it will only trap under such conditions. So what is going to happen? Let us imagine that light from, which is falling on this half of the bead is much more intense than what is falling on this half of the bead. Simply because this is the more intense region, this is on the other hand the less intense region. Now this light is going to fall on it and get refracted in this particular way and move out. So what is happening is that the light would have, if the bead had not been there, it would have obviously gone off like this, right? However, it is now moving out like this, so there is a momentum change which is associated with changing the light direction from this to this. And this is the momentum change. There's going to be a recoil which is going to act like this. So this is the arrow on this particular bead. Now, the same thing is going to happen on the other side. However, it is less intense, and thereby the momentum change that happens in this direction is going to be much smaller than what is happening in this direction. The bead is also going to suffer a recoil from this, and that is essentially shown by this. Okay, so it is opposite to this particular arrow. Now, if I could resolve this, this is what physicists would do or mathematicians would do. You find that the force is more strong, is acting on the bead finally towards the more intense regions. So this bead is going to feel a force towards the more intense regions. So essentially it is going to get trapped in regions of the laser where the intensity is the strongest. Right? So now that we understand this, just a small, because I find this extremely inspiring. Arthur Ashkin was awarded the Nobel Prize in Physics in 2018. Okay, and it was given for groundbreaking inventions in the field of laser physics. And half of that prize, it was actually shared by 3 people, and half of that prize went to Ashkin for optical tweezers and their application to biological systems. Now, this is the paper which came out in actually 1986, which had the first working optical tweezer. And in that you can see he basically trapped particles from 10 micrometers to 25 nanometers in water. Now what I want you to see is the next slide, okay. So these are the progression of papers which were published on the same area. 70, if you remember the figures that I showed you, 1970 was the first attempt at the optical tweezer, and 86, 16 years of effort when the first working tweezer came out. And in 18 was when, at the age of 96, that he received the Nobel Prize. So what is a very important message for all of us is that if you really believe in something passionately, never give up, okay? So I think it's a very, very inspiring tale for us. So now let us ask the question that, okay, you have trapped a small particle, what are you going to do with it, all right? So the first thing I would like you to understand is that when there are small particles suspended in water, this is what you would see, okay, a Brownian movement, all right, of the particle. Now the question is, how do you actually qualify this Brownian movement? This is happening because this is the particle, it's constantly being bombarded by all the, water molecules. Let us say it's suspended in water by the water molecules about it. So it is essentially doing what we call as a random walk. So these are certain trajectories that you can see, which are happening. It's a random walk. However, there is some, this, for instance, is a very important thing. It tells you the probability of a certain amplitude, okay. So for instance, it's hit, it moves a certain distance. So that amplitude, what is the probability of that amplitude? And you can imagine that as the amplitude gets larger, you're going to have the probability also fall down. So basically, larger fluctuations happen at lesser frequency, all right. Now this over here is something that we called a power spectral density. And a power spectral density essentially tells you what is the, the energy content at different frequencies, all right. And this basically, in the case of a pure Brownian motion, will follow a 1 by f square kind of a behavior, okay. So let's not get into too much detail over here, but basically you have to understand that basically this power spectral density kind of falls off, and it falls off with a slope of -2 for the frequency, right. Now this is the case of a free Brownian motion. But now what do we have? We have a particle which is sitting inside a trap. So obviously it is not an unrestricted Brownian motion. So let us see what happens in that case. So you have this particle over here, and it is trying to undergo Brownian motion. When it does small little fluctuations, it is exactly the same as Brownian motion, all right, which is unrestricted. However, if it tries to do large fluctuations from this point, it is going to start feeling the fact that it is going to be it is in a trap. So to put it in a very trivial example, a trivial analogy, your leopard which was in a cage, if it just does small perambulations in the cage, it may not even realize that it is in a cage. But suppose it really wants to run, okay, in a particular direction, it knows then its behavior is going to be modified. Why? Because it is actually confined, right? So this is precisely what is happening. It was a very trivial example, don't take it too far, but nonetheless, this is how we can understand that small fluctuations are going to leave the bead behavior unaffected. However, when it tries to do very large fluctuations, it is going to feel the effect of the trap. Now, let us ask the question, what is the frequency with which the small fluctuations will happen? They are going to happen at very high frequencies, right? More small fluctuations, and as I showed you with that probability, large fluctuations are going to happen at smaller, frequency. So thereby, only when I go to very large fluctuations, which is going to happen at the small frequency end, I'm going to see a change in the behavior. So now let me just show you the next one. Okay, next slide. Okay, it has changed. Okay. So this is the same power spectral density which I showed you, and if you saw it beyond, at the high frequency range, you would say, hey, it's still a 1 by f square kind of a fall off, okay. So this is not any different from the free Brownian motion. However, when you go to the low frequency end, you start seeing an effect of the trap, okay, and you see that it has been modified. So this is what we call as the modified Brownian motion, and essentially this frequency at which there is a crossover from the free movement to the trap defined movement, okay, is what we call as the corner frequency. And this in some sense tells us about the strength of our trap. Okay, so how strong is the trap? Okay, so it's a way of characterizing our trap. So instead of a 1 by f square kind of a free fall off, you actually have a fall off which is like this, 1 by f square plus this corner frequency square, and this corner frequency for us is a measure of our trap strength. Okay, so now move. Sorry. Okay, so this is, so the power spectral density as we see is that any kind of dynamics that you see, you will try to see what is the energy content at various frequencies. So now in our case, what do we have? The energies are associated with fluctuations, right? So the smaller fluctuations actually will happen with very high frequency. So they are actually sitting at some end like this, and the larger fluctuations are going to happen at smaller frequencies, okay? So what is happening in the case of free Brownian diffusion, you will see the straight line just continue, okay? However, here what is happening, larger fluctuations are being stopped by the trap. So you see essentially a modification, okay? Instead of continuing like this, what has happened? The curve is actually, the experimental curve is actually turned off like this. So basically, this frequency which you have over here, somewhere roughly over here, actually you would fit it to this kind of a formula. This frequency over here signifies the change from free Brownian motion to trap Brownian motion, and it's a measure of your trap strength, okay? So this Fc over here, which we say, with other factors of course, but it is an estimate of your trap strength. Is that okay? So let's move on now. So, okay, I think this is a kind of a repetition of the same thing, except let me just point out to you that the corner frequency, as I said, is an estimate of your trap strength. Your K is actually your trap strength. But there is also this factor where gamma is some kind of a friction coefficient. Now, this depends upon the fluid you're surrounding the particle with. So, if you surround it with water, it will be one thing. If you surround it with glycerine, it will be something else. Okay, but you could also put something which is even more complicated. Okay, so I will come to that now. Right. So, let us imagine now you have put in a particle which you understand very well. So, very often you just go, you know, commercially you can get beads which are made out of plastic, so polystyrene beads which have a fixed diameter, they come well characterized. So, essentially you know that this gamma which I showed you in the earlier slide would have a factor like this, 6 pi eta r, r being the radius of that sphere, and this eta which I am showing over here is essentially to do with the viscosity of the fluid around it, alright. So now if you know the r, then you could basically, you know, if you know both the r and you know that you are putting it in water, so you know the viscosity, you can essentially, and you have measured the corner frequency, you can get a gauge of the trap stiffness. Okay, so now your trap is well characterized. Once your trap is well characterized, you can perhaps go and study other things. So now, if your trap is characterized, all right, so you know the K, you know the bead that you're using, what you can now start doing is changing the fluid around it, all right, and start studying the suspension fluid which is around your trap particle. So let me just show you what you could do. Okay, I think I missed a slide. So basically, this is where it becomes very interesting to people who study soft matter, because we know that it's not a simple liquid, all right, and depending on the shear rate, so this is what I have put in the next slide. So some examples are given over here. I will not spend too much time. So this is a strict classification of a, you know, Hookean solid, which has a wonderful stress-strain relation. This is a Newtonian fluid which has a constant viscosity no matter what the shear rate. But then you have a whole bunch of materials, especially most of our day-to-day materials, which actually have a viscosity which depends upon the shear rate. Okay, so you could have shear thinning, shear thickening liquids, and all such materials you could actually study in what sometimes is called as microrheology in an optical tweezer. Okay, so let me not get into all of this. What I've just tried to say, the soft materials are complicated and living materials, okay, are perhaps the the most complicated simply because they are self-assembled, they are out of equilibrium. Just to maintain being the way they are, they need to consume energy, okay? So from a physicist's point of view, living materials are very complicated, and trying to understand them is indeed a very big challenge. So here I've given an example of what kind of suspensions you could have. So for instance, in our lab in Bangalore University, people have actually studied active suspensions, which is essentially a bacterial suspension, which are highly complicated material to study. And then there has been also studies on regenerated silk fibroin. This is some certain scanning electron micrographs which I'm showing. And also there have been studies which have been done on ferrofluids, which are suspension of nano-sized magnetic particles in a liquid, and they also have very, very interesting behavior. But we will go on a bit further because I wanted to tell you about biological applications. So see, these are certain kinds of applications which I've picked up from other papers. So this is from essentially these people. And here, everything is being done, as you notice, it's being done with a single trapped bead. Now, what has been done over here is that they have taken a DNA strand, all right, and these are essentially the nucleosomes. And I think there are a lot of biology people over here, so far be it from me to kind of explain to you what a nucleosome is. But for us physicists, nucleosomes are essentially about 150 base pairs of DNA wrapped around some 8 proteins which are called as histones, if I'm not. So it's kind of a unit, and essentially they arrange themselves in this particular way. Now what has been done over here is that this strand of chromatin has been attached to a cover slip over here, and the other strand has been attached to a bead which is trapped. Now what is being done over here now is that the cover slip is being made to move, right? And what you get essentially is a force extension kind of a graph. And what you can see in the low force range, there is an extension. And then at the high force range, what you start seeing is this sharp jagged peaks. And each of these represent what is called as a disruption event, which is basically the unfolding of a nucleosome. Okay, it's basically unraveling. And if you can count the number of disruption peaks over here, it basically kind of tells you the number of nucleosome core particles which were sitting on this entire strand. Now, this force extension curve has been done also for a naked DNA which had no nucleosomes, and that actually should have looked like this. So you can see how different it is, and it is a way of studying what is really there on a DNA strand in a very different way using just mechanical force. So now let me go to another example. So these are live cell experiments, and here you have this, actually 2 experiments have been kind of described over here. Here you have a cell, all right, and there by a ligand binding on a, so you can basically have the microbead bead that you're trapping coated with certain kind of ligands which will essentially bind to a cell membrane. Now what you can do is essentially you start the trap movement, so the trap starts moving away, and at some point, of course, this is going to give up, all right. The strength of the bond is not going to be strong enough, and it is going to basically disrupt. So this is what you see over here. This is again a force distance curve. You can see that this is the extension, and then you finally have a flattening. So this is where basically the ligand strength, bond has given up. Now what is interesting over here is that there are of course different factors which are going to contribute to when it is going to snap. And you could actually change those characteristics, the suspension fluid, the chemical environment, and so on and so forth, and see where does this actually happen in each of those conditions. So this is another way, interesting way in which you could study how strong these particular bonds are. The other thing that could be done is essentially over here, this is another experiment which has been just shown in the same slide. You could have a bead which is basically pulling a part of the membrane along with it. So this is basically a tether which is formed, and the tether is being pulled away. The trap is moved in this particular direction. And this is the force distance curve which has come out of this. And you can see that this point is where the tether is actually forming. And then you can see that this is a place where it has actually given up. Okay, the tether is disjointed from the trap. And this particular one tells you about 3 such events. Okay, so there were actually 3 tethers which formed and they got disrupted at 3 different points. Okay? So this is yet another way in which, live cells could be studied, and what is their bonding strength with other, other chemicals and so on and so forth. Here you have now not just a single trap, you have a dual trap, and this is basically where you have a trapped bead in this first optical trap, which has been attached to a DNA, and here you have another one which is which is a moving trap, and the DNA is is been tethered, or sorry, I should say attached to the bead over here. And then you have this trap which is being moved. Now remember, the movement is done in a very calibrated, in a very controlled fashion in all of the experiments that I'm telling you. And what you see over here is basically the behavior when you get into what is called as an overstretching transition. Now this overstretching transition, what does it mean? So each of this particular DNA, I believe it has been taken from a phage which attacks the E. coli bacteria, okay, and this has a contour length of about 16 micrometers. So contour length, I guess, is the length, the maximum length to which a DNA molecule. So if you kind of add up the lengths of the individual components, that would be basically the contour length. And when it has been pulled beyond the contour length, as you can see, of 16 micrometers, you start seeing the overstretching transition, and basically the DNA will now disintegrate. Okay. Now this particular behavior again depends upon the different environmental conditions, and you can see when this disruption event will actually happen under what kind of chemical environment. So this is another way in which you could study. And let me show yet another application, and this is again to do with red blood cells. I'll be returning to red blood cells in the latter part of my talk. And this is again a red blood cell. So there we had a DNA which was attached with 2 beads sitting in 2 different traps. Here you have a red blood cell which has been attached to 2 beads, and the 2 beads are sitting in the 2 different traps. Now what is done over here is not just a lateral motion of the second trap, but rather an oscillation. So a to and fro motion of the trap. Now what you see over here, so this is just a top view of the whole thing. So this is your red blood cell, and these are your 2 beads, and they can be, this bead can be essentially shifting between this position and that position, and this has been done at the frequency of 200 hertz. So 200 times it will be essentially moving from doing a to and fro motion in a second. So what you see over here, the red curve, okay, is essentially following the motion of the bead, of the trap, the signal that you have put on the bead to make the movement happen. So this is essentially going up and down at 200 hertz, all right. So this is the time axis, this is your position. Now what you see over here in dark is essentially the behavior of the bead which is sitting in that particular trap, and you can see it almost follows the red line, okay, it almost follows the red line, okay. So it is a little out of phase, but it is almost following the red line. And the dotted line that you see over here is essentially the movement of this bead. Now why is the dotted line not moving in the same phase as the signal or the first bead? Simply because the movement that it has to feel has to be now transmitted through the red blood cell, and thereby the mechanical properties of the red blood cell, how elastic is it, is there some information being lost, the mechanical motion information being lost by the time it comes to this bead, it all comes into play, and thereby you see a phase lag, the amplitude is much smaller. So hidden in this particular information, in this particular curve, is the information about the viscoelastic behavior of the red blood cell. So this is again something you could do with an optical tweezer to figure out about the mechanical properties of, let's say, a human red blood cell, right? So with this, I think you can, of course, go to more complicated, the trapped object could be even more complicated. It need not be a sphere. So for instance, there are experiments that people have done with trapping, let's say, more than one sphere in an optical trap, or, working with something like this. Professor Pomorski showed us the echinocytes in the morning. So you could take some such thing And then instead of, on your power spectral density, you would also have these peaks which would be riding on top, okay, because this goes into some kind of a rotatory motion inside the optical tweezer. And there is interesting information to be got out of such kind of signals. So, yeah, so let us just see whether this works, this particular video. I'm not sure it will, but let me give it a go. This is an avian red blood cell, okay, in an optical tweezer. Now, the avian red blood cell, I should have shown this perhaps after the red blood cell behavior. You can see the dynamics. It actually does a dual flip. So Chetna, who's with us, she has actually done a lot of work on this particular motion of the chicken red blood cell. And if any of you are interested, I'm sure you can talk to her. And they really have fascinating dynamics. Now, the dynamics of the red blood cell, the human red blood cell, is not as interesting as this one, but more easy to understand from a physicist's point of view. And what we will be doing in the second part of the talk is at least to understand how we can get membrane stiffness information from that kind of a dynamics in the optical tweezer. So do we take a break now, or? Okay. And then I come back after this. Thank you. Yeah, I'd be very happy to answer any questions if there are any. Okay, so that is a very complicated thing. I do have the papers from which I've taken it. I will share it with you, okay? I personally don't do that, so I really don't know how exactly they are attached to the beads. Sorry? The bead is, okay, so are you asking about the mechanism by which it is being trapped? So the point is that the beads are, these are like plastic beads or polystyrene beads, or sometimes even silica beads. They are like, commercially manufactured, and they can come to you with different kinds of coatings, okay? So you can ask the, the manufacturer to give it to you with a particular kind of coating, and that coating will depend upon what particular application you want it for. So for instance, if you want it to attach to a DNA, you will ask for a particular kind of coating which will help you attach the DNA to that. Now, if you ask me the question as to how do I prevent it from attaching with more than one DNA, or how is it, so this, what you have seen, is an extremely simplified picture. So for instance, that picture which I showed you about the cell membrane tether being drawn, actually the cartoon showed only a single tether being drawn. But when you saw the force extension curve, what you see is that there are actually more than, there are 3 disruption events, right? There are 3 points in which, so actually 3 tethers had been joined. So such things are bound to happen. You will not be able to ensure only a single DNA joining, but then you'll have to do multiple analysis of your work and then see whether you can figure out which one was a single event and do the entire analysis on basis of that. Okay, I think that is how it normally works. It will not work as simply as been shown in the cartoon. But I don't know whether I have answered your question. Okay, the beads need to be transparent, yes. And as I told you, the refractive index of the bead has to be higher than the surroundings. Otherwise, the refraction will not work the way I showed it to you. That is definitely required, which is why you can use, polystyrene or silica if you are suspending it in water. Sorry? Absolutely, absolutely. In fact, what I will show you, so for instance, this last video that you saw, it is actually the chicken red blood cell which has been trapped in the bead, okay, in the optical trap. And what I will show you after this is what happens when you actually trap human red blood cells, okay. So definitely you can trap vesicles, yeah. The power spectral density is what you're talking about, yes. Yes, yes. You do a Fourier transform basically, yeah. Okay,
So the point is, the question that you can ask is that, you know, what kind of noise am I likely to get? So the thing is that, of course, you'll have to, you know, make sure that all your equipment is very well grounded, okay, for one, because electrical noise can be, anything that is at 50 hertz, you better ignore because that's coming from your AC. These experiments are all done on vibration isolation tables, so thereby you wouldn't have that kind of vibration. Apart from that, I don't think we do anything more special, okay, to get rid of the noise. So this is what I kind of recall. We don't do anything further. Yeah, that's the thing. Yeah, please go on. Okay, so do you want me to f square plus fc square? Yeah, yeah. So I'll just get back to this. So let me just, this is a slide I kind of jumped over. So what you can see over here, okay, this is basically your frequency axis. So it's of course in a logarithmic scale. And this is essentially your, the power spectral density as we call it, alright. Now what you would essentially do is, whatever you just said, so since I kind of skipped over this, you got it. What you essentially do is you measure the position as a function of time. Alright, I kind of skipped over this entire thing. And then this, of course, is in the time axis. It's how it's behaving in the time axis. A Fourier transform of this is done, and then you get it in the frequency domain. So you get the frequency, and this is your power spectral density. Now, what you should have had, if it was maybe the earlier slide has demonstrated this better. If it was a pure, let me go back one slide further. So here you have basically a power spectral density, okay, of a free Brownian particle. Now just note that you have again the logarithm of the frequency over here. Please note that the power spectral density, it follows a 1 by f square at high frequencies as well as low frequencies. There is absolutely no change. Right? Now, when you come to a trapped particle, this is what you measure. The colorful graph, that is what you measure. So what you are noticing is that the behavior is essentially modified below a certain frequency. So the way I kind of have explained it right now is that what is really getting affected. So the Brownian motion, of course, there is going to be fluctuations, there are going to be small fluctuations, there are also going to be large fluctuations. When the particle is doing a small fluctuation, it basically doesn't even sense the trap. Okay? So the behavior is completely like that of a free particle, free Brownian particle. Now, what is the frequency at which small fluctuations are going to happen? They're going to happen at large frequencies. Right? You can see a lot of events which have very small fluctuations. So essentially, that is this part of it, the high frequency. So then you can see a 1 by f square kind of order. Now, what is happening over here is that you are now basically trying to see the number of events which are large fluctuations. Now, it's at large fluctuations that the influence of the trap is being felt. So, as a result, what is happening is that your power spectral density is getting modified, and it should have actually gone like this if it were a free particle, this straight line that you see over here. However, it has gotten modified and flattened over here. So now what you could do is you can fit it to a graph like this. So please note, instead of 1 by f square, you have now a 1 by f square plus fc square. So this fc is now going to give you a gauge at where this transition into feeling the influence of the trap is going to be found. So you can well imagine that you had a very, very strong trap. Okay? Even small fluctuations may start getting confined, right? So this is going to move further away. So which means it will give you a larger value of Fc. So that's why I say the Fc is a measure of the strength of the trap surface. Okay? So, yeah. So that is this. Any other questions? Yeah.
So now during the tubular experiment, so there is a point where the forces are in equilibrium. So that is the point where
So basically what you're doing is that you're trapping the bead, okay. So yeah, your forces are in equilibrium. Now what you're doing is that, now with this bead, what you can do is that you can apply very controlled, measured forces on other things. So what you're doing is that with this bead and the chemical, way you've modified its surface, you're able to make it attach to a cell and pull a tether. So how are you pulling the tether? You are actually moving the optical trap itself away with the bead, okay. So the cell is in one place, the tether has been pulled, and you are now observing the behavior of the bead and seeing how far you can go till there is a release, and when that releases, you can actually see a change in the behavior of the bead itself. So remember, in all those events, it's the bead that is trapped. The bead is then attached to something else that you are interested in. It could be a cell, it could be, let us say a DNA strand, it could be, what else did we say, red blood cell, okay. And you are basically observing the behavior of the bead itself, okay. You're not really observing the behavior of the cell. You could be looking at it under a microscope, but what you're seeing is the dynamics of the bead now. So obviously when the trap is moving away, the bead is not moving that easily with the trap because it is attached to the cell, right, through this tether. And at some point it will revert back to its original behavior, at which point you can figure out that the tether has been moved away. You know the speed at which you have been moving the, the entire trap, okay, the stage which, how fast it has been moving. And from that you can calculate exactly at which point that detachment happened. And that will in some sense tell you about the strength or with which you had been, the tether was attached to the bead. Okay, so this is, so remember the bead is the one which is trapped. Okay, over here nothing else has been shown except, as I told you in the last slide, where you had actually trapped an avian cell, avian red blood cell, yeah, to be more specific. Yeah, any other questions? Mm-hmm. Mm-hmm. So just 1 second, let me just get to the particular. Huh. This is, you're talking about the cell, right? Yeah, so this is, yeah, the cells, it's always in piconewtons, and this one you're talking about, it's in piconewtons, and that is in micrometers, yeah. Cell, actually, it was the frequency which we were looking at. This one, right? Another one. Okay. So this is, of course, micrometers and piconewtons. Let me see which one you're talking about. Nanometers, I don't think so. Ah, yeah, okay, this is the extension, but please note, it's in thousands of nanometers, which makes it in micrometers, okay? Yeah, so it is still micrometers, I would say. I really wouldn't, I think it's, yeah, so it is like 0.8 kind of micrometer or so, it's still in hundreds of nanometers. Yeah, so it is kind of still happening at less than a micrometer, but not that much less than a micrometer. Yeah, so I think it is wrong to say nanometers, but it's what you have to understand is the resolution. You can see over here, right? So the fact that you could actually count the number of disruption. So these are happening, the distance between each of these is in nanometers, okay? So that is the point. So it is really a fantastic experiment in my opinion. All right, so each of these, you can see the extensions are, between them, they are really in tens of nanometers, I would say. Yeah. Okay, any further questions?